Hi friends, once again welcome back to my channel. Today we will discuss T helper 2 response. That is, we have already discussed T helper 1 response in the previous classes. That is, the T cells, that is T helper cells, they are having two types of responses, or they are exhibiting two types of responses. They are T helper 1 response and T helper 2 response. In helper 1 response, cytokines are produced to activate a group of cells, mainly T cytotoxic cells, natural killer cells, macrophages, eosinophils, neutrophils, etc. In helper 2 response, cytokines are produced for the activation of B cells, and these B cells get activated and they start proliferating to form two types of cells. They are plasma cells and memory cells. Plasma cells, they are secreting antibodies, and memory cells are providing immunological memory. The first step in T helper 2 response is internalization of an antigen by receptor-mediated endocytosis process. That is, the antigen is internalized by the antigen-presenting cell. Antigen-presenting cell may be a macrophage, dendritic cell, B cell, etc. And these antigens are processed inside the antigen-presenting cell into peptides. An antigen binding, it initiates a signal for the expression of class 2 MHC molecules and B7 molecules on the surface of antigen-presenting cell. And this increased expression of class 2 and B7 molecules enhances the antigen presentation ability of the antigen-presenting cells. The processed peptides are then presented on the surface of this antigen-presenting cell along with class 2 MHC molecules. This is because T helper 2 cells, these are class 2 MHC restricted. This peptide MHC complex is ready to induce a T helper cell activation. Then we can see several steps or stages in T helper cell activation. In the first step, antigen-antibody complex, it is internalized by receptor-mediated endocytosis. Here we can see, and this antigen, it is degraded into peptides, and this peptide, it is displayed along with class 2 MHC molecule on the surface of antigen-presenting cell. The next step, the T helper cell in the circulation, it will come and recognize this antigen which is presented along with class 2 MHC molecules. So this is the B cell or an antigen presenting cell, and it is displaying this antigen on the surface with the help of class 2 MHC molecules, and this T helper cell, it come and recognize this antigen. And this is the second step, that is T helper cell recognizes the antigen class 2 MHC complex present on the surface of antigen presenting cell or B cell. In the third step, after the interaction between the antigen and this T helper cell, we can see they are forming a trimolecular complex, and the third step is the interaction of CD40 with CD40 ligand. That is, CD40 is present on the surface of B cell or the antigen presenting cell, and CD40 ligand is present on the surface of T helper cell. They interact and produces a signal. Then the next step, the B7 molecules present on the surface of these B cells and CD28 present on the surface of T helper cells, they provide co-stimulation. That is, they also interact to produce a co-stimulation. The next step, B cells, they begin to express receptors for various cytokines. That is, after these interactions, we can see B cells, they start secreting or expressing receptors on their surface for cytokines. As a result, the T cells, they start producing cytokines, and these cytokines, they come and bind to the surface of these B cells. Binding of these cytokines released from the T helper cells, it sends a signal, and this support the progression of B cell to DNA synthesis, and ultimate result is the differentiation of B cell, and the B cell undergo proliferation. And some of the activated B cells, they become memory cells, and while others get converted or transformed to plasma cells. Plasma cells are antibody secreting cells, and they start secreting antibodies, and the first antibody produced is IgM. And the memory cells, they remain in circulation and provide immunological memory, and they can mediate the secondary immune response to subsequent contact with the antigen. That's all about T helper 2 response. Hope you all understand the session. Thank you. Have a nice day.